Hello student, good morning. Today I have start the new topic, that is the Mendel's law of inheritance and its extension. Mendel's law of inheritance and its extension. The Australian monk Gregor Mendel is considered as the pioneer of modern genetics. He was born in July 1822 in Hinzendorf in Australian Silesia. He graduated from gymnasium in 1840. In his youth, he led a disastrous, poor, difficult, and sad life. During 1854 to 1868, Mendel taught physics and natural sciences in a higher secondary school of Broome, and besides teaching performed certain hybridization experiments on fruits, trees, flowers, vegetables, and more especially on gardens. Peas. He published his conclusion in 1868 in the annual proceedings of the Natural History Society of Broome. Unfortunately, his remarkable piece of work remained unattended for many years, probably because the biologists of that time were busy in removing the confusions and controversies which might have been arisen by the publications of Darwin's Origin of Species. It might also be possible that biologists of his time could not understand his statistical approach and could not appreciate his work. It was in the beginning of 20th century that three botanists, namely Hugo de Vries of Holland, working on Enothera, Karl Corens of Germany, working on Xenia, peas and maize, and Erich von Schirmack of Australia, working on various flowering plants. Independently drawn, the conclusion, like Mendel later, these botanists came across the research paper of Mendel and rediscovered it in 1900. Mendel's original paper was republished in Flora 89 364 in 1901. Batson confirmed Mendel's work by a series of hybridization experiments. Mendel's considerations for his hybridization experiments Mendel had certain considerations in his mind about the choice of a suitable material. Mendel's consideration about the material were as follows. Number one, variations. The organisms which are to be chosen for the genetical experiment should have a number of detectable differences, and at a time only single detectable characters should be considered. Second is reproduction. First is variation, second is reproduction. That type of chosen material for the experiments. The chosen organisms should be sexually reproducing, that is by fusion of male and female sex cells, because only then the offsprings will be able to receive different characters from both the male and female parents. Third, control mating. The chosen organism should be able to mate in controlled or well planned conditions, because in genetical experiments sometimes we have to near genetically pure parents by methods of control mating. One should maintain careful records of the offsprings of many generations. Fourth number, short life cycle. That material chosen the short life cycle. The chosen organisms choose have very short life cycles, should have very short life cycles. Fifth number, large number of offsprings. Large number of offsprings means next generation, many number of generations. The organisms which have been chosen for the genetical experiments should produce large number of offsprings after each successive mating, because it will help in deducing the correct conclusions. Sixth number, convenience in handling. The experiment species should be of a type that can be raised and maintained conveniently and inexpensively in the laboratory. For instance, the elephant will prove entirely useless material for genetical experiment. Then the Drosophila, pea plants, tomato, rats, guinea pig, etc., which have been generally used and are still used in the hybridization experiments. Self-fertilization of flowers, Pisum sativum, portion of the keel which encloses reproductive organs, has been cut open to show the stamen and stigma, the mature fruit pod containing seeds that develop from the fertilized pea flowers. Mendel's material. Mendel's found edible pea, Pisum sativum, that is the botanical name, a best material for his hybridization experiments. The pea plant has various contrasting characters among its different varieties, such as stem may be tall or dwarf. Stem may be tall or dwarf. Cotyledons may be green or yellow. Seeds may be round or wrinkled. Seed coat may be colored or colorless. The Unripe pods may be green or yellow. The ripe pods may be inflated or constricted between the seeds. Flowers may have axial or terminal positions, and the colors or terminal positions and the colors of flowers may be red or white. Besides these contrasting characters, the pea plant is a very satisfactory material for the hybridization experiments due to its flower structure. The flowers of pea plants are so constructed that the pollens of a flower normally fall on the stigma of the same flowers and thus effects self-pollination or self-fertilization. Mendel's easily prevented self-fertilization by removing the stamens from the flower's bud, because for genetical experiments the cross fertilization was the fundamental necessity. So Mendel cross pollinated two desired pea plants differing in a particular pair of contrasting characters by placing the pollens of the one flower on the stigma of other flower whose stamens have already been removed, further to prevent artificially pollinated flowers from the contamination of pollens of unknown source by insects. Mendel covered the flowers by the bags. Mendel's procedure for getting the exact results in the breeding experiments. It was necessary for Mendel to rear genetically pure variety of pea plants. For a single characters, Mendel adopted self-fertilization technique. For instance, to get pure characters for tallness, he self-fertilization a tall pea plant for many generations till the resulted offsprings always produce only tall plants. Likewise, he got genetically pure variety. For dwarf pea plants, Mendel's cross pollinated these two varieties of pea plants, which were differing in a pair of contrasting characters, tallness and dwarfness of the stem. When he made observations on the offsprings of first generation, he found only tall plants. He allowed the self-pollination in the offsprings of first generation and made further observations on the offsprings of the second generation. It was astonished to note both tall and dwarf offsprings in the second generation. This showed him that characters of dwarfness disappears in the first generation, but again reappear in the second generation. Further, the tall and dwarf plants of second generation were always in the ratio of 3:1, 3 tall and 1 dwarf. He self-pollination the dwarf offsprings of the second generation and found only dwarf plants of the third generation. But when he self-pollination the tall plants of second generation, then he found that 1/3 tall plants yield only tall plants in third generation, while rest 2/3 tall plants yield tall and dwarf plants in the ratio of 3:1 on the basis of the results of his experiments. Mendel formulated following three laws: law of dominance, law of segregation, law of independent assortment. Actually, Mendel himself did not postulate any gene principles or laws of simply gave conclusion, theoretical and statistical explanations for his hybridizations. Experiments in his research paper, however, it was Correns, the discoverer of Mendel's work, who thought that Mendel's discovery could be represented by these fundamental laws of heredity. Example: parents tall cross with dwarf. Genotypes: capital TT, dwarf मध्ये small tt. Homozygous. Types of gametes: capital T, single capital T and single small t. F1 generation: capital T, small t. Hybrid tall, monohybrid. Type of gametes: T, capital T, small t. F2 capital T, small t, capital T, small t. TT tall, capital T, small t tall, capital T, small t tall, and both small t, that is the dwarf. Means phenotypic ratio is 3:1, 3 talls and 1 dwarf. Genotypic ratio 1 pure tall, capital T, 2 hybrid tall, capital T, small t type 2, and small t, that is the pure dwarf. Genotypic ratio is 1:2:1. This is the monohybrid ratio. Law of dominance. Okay, thank you.